All right. Well, it's 5 after, so let's go ahead and get started. I'll give people a few more minutes to come in while we're doing introductions and some light housekeeping. So again, my name is Wiley Hall. I'm the Director of Research Chemistry with Safe Food Alliance. Our speaker today is Elise Palmer, who I'll introduce in a minute. So while she's going through her webinar, I'll be manning both the Q&A and chat, along with our own internal tech support. So please, if you have any questions, feel free to submit them using either the chat or preferably the Q&A. If it's something that I think needs an immediate answer, a clarification on a point, something like that, then I'll go ahead and maybe break into Elise's talk. Otherwise, I'll add it to our list of questions to be asked at the end of the session. All right. So let's go ahead and get started. So our speaker today is Elise Palmer. Elise is a research chemist and a key member of the Vicam Waters Business R&D team. She's followed a strong scientific career in both analytical and diagnostic technologies that's included 7 years of product development roles for various companies at the forefront of food and agricultural safety, and 7 years in academic medical research. She joined Vicam in 2013 and has been instrumental in the development and improvement of several solutions for customers in the field of rapid diagnostics. Elise holds 2 degrees, an accelerated undergraduate master's in biochemistry from the University of Bath in the UK, and a master's in business administration from the University of Rochester in New York. Elise, today's title of today's talk is Rapid Mycotoxin Detection Technologies for Meeting Regulations, and please take it away.
Hi everyone, welcome. Thank you for logging on to listen to me today. I'm speaking to you from Massachusetts, so it's afternoon here, so I will say good afternoon. And with that, let's get started. Let's try and get things moving. There we go. So just to give you a brief overview of what we're going to cover today, we're going to talk about why should you care about mycotoxins? Why are you listening to this? And what might happen if your product is affected and it reaches its market, and what can you do to protect your product and your business? So briefly, why should you listen to me? As Wiley said, I'm part of the research and development team for Vicam. We're part of the Waters Corporation business. Vicam was created back in the early 80s. It's been around about as long as I have. Their first product was launched in 1985. That was the Aflatest column, which we'll hear a bit more about later today. It's still going strong. In the mid-2000s, we joined Waters, and we've been moving things along ever since. So a brief introduction to mycotoxins. So normally, if I'm doing this live, I would ask the audience if you knew what a mycotoxin is, if you could name any common ones and any guesses. But since I can't do that today, I'm just going to tell you. So I find this picture fascinating. There's something very odd going on. Like some guy seems to have a lampshade on his head, another guy maybe a pair of underpants or a napkin. They've got foxes' tails pinned to their clothes. It's like, what is going on? There's limbs missing. This is a picture from the Middle Ages, and these gentlemen are suffering from something that was called then St. Anthony's fire, and which we now know as ergotism, which is a form of mycotoxicosis that has some pretty dire consequences. Some people believe it was implicated in the Salem witch trials here in Massachusetts, for example, although that's a little controversial, but some of the symptoms can include gangrene, psychosis, tremors, itching, like all sorts of horrible, horrible things. So this is just to show you that the problem of mycotoxins has been around forever. It's kind of grown with us through time. But the modern story really begins in the 1960s. There was a case in the UK, where I'm from, of something that they called Turkey X syndrome, where a whole flock of turkey poults succumbed to a strange illness, and it was eventually traced back to their feed, and what they had suffered was a form of aflatoxicosis, and the term aflatoxin was coined. And since then we've been really studying mycotoxins and regulating them and developing testing regimens for them. But that doesn't mean that the problem has gone away. Here we can see 3 examples. Back in, I think it was 2004, out in Iowa, a number of companion animals unfortunately died due to contaminated pet food. Out in Africa and Kenya, there's an ongoing problem with aflatoxins, and that's simply due to the system, production and storage system that exists there. It makes testing regimens very difficult to implement, and it has a big impact on the community. You know, aflatoxin is the most carcinogenic naturally occurring substance known to man, so you can imagine small children are particularly vulnerable to that. And the bottom image you can see here is some cows. Back in the early 2000s, again, there was a crisis with contaminated milk coming from the Balkans in Eastern Europe, contaminated with aflatoxin M1 that went into products in Western Europe that were then sold throughout the world, and it resulted in a near political breakdown between Western Europe and Russia. In fact, it was a very serious situation. So we know that mycotoxins are bad. They have bad consequences. We want to keep them out of the food chain, but what actually are they? Well, they are the secondary metabolites of various fungi. Some of the most common ones that are tested for, or that we should be considering, are listed on this slide. The most commonly tested for are highlighted in blue. Those in yellow are currently not regulated, but may be tested for. And so you may have heard of aflatoxin that I mentioned. This is primarily produced by Aspergillus flavus and Parasiticus species of fungus. And it can be found in a number of different things. The most common that people think of would be something like peanuts or tree nuts, but it can also be found in grains. Another that we should consider perhaps more seriously than we currently do is ochratoxin A. This is produced by another couple of Aspergillus species of fungus. And it's found in a wide variety of different commodities, including things such as raisins, going through barley, soy, and coffee. And while it might be found at very low levels in those items, those are things that feature quite heavily in our diet. So we have to consider that a low level of contamination could still be serious for human health, because it can bioaccumulate in the blood and tissues of the body. So a couple of others, fumonisins, are produced by Fusarium species. That's something that we would primarily be focusing on grains. T2, HT2 are actually listed as a weapon of warfare, I believe. We have a lot of trouble getting standards for T2, HT2 into the lab, and when we're manufacturing things with that, we have to sign all sorts of strange paperwork. It has such dire health consequences. Deoxynivalenol here, its other name is vomitoxin or vomit toxin. I think that's pretty self-explanatory there. It's something you don't want to find in your beer, which a lot of us have been relying quite heavily on that beer during this pandemic just to get us through, along with the coffee. So we've got to really consider that DON and that ochratoxin. And zearalenone is another that can be found in grains. And it has implications, particularly in animal health. It can be a concern for breeding because it can lead to problems with infertility and some spontaneous abortion, things like that. So we know that they're bad, and in fact, the most important chronic dietary risk factor, so perhaps more harmful than a lot of synthetic contaminants that we might be looking for, plant toxins, pesticide residues, food additives, things like that, that we might also be testing for. So, mycotoxins often get pushed to the side. It's like, oh, there's only a few and you have to do a couple of tests. Don't really have to worry about these. Let's focus on the big ones. But actually, we can't forget about them because the consequences can be very, very serious. So how do we avoid them? what are some of the ecological parameters that affect mycotoxins? So as I mentioned, they're the secondary metabolites of certain fungi or mold species. So primarily what you want to do is avoid growing those molds. But it's not as simple as saying, okay, this thing is moldy, I'm going to throw that away and everything else should be good. Sometimes the mycotoxin can be present and the mold is not. So it was, when you got rid of it, mycotoxin remains. So those factors that we want to eliminate to prevent the production of the mycotoxins would include things like controlling the moisture, the spore load of the fungus that's present, so minimizing that, the temperature. If there's any other contaminating bugs, be they good or bad, on the product, the competition of the fungus with that microflora can induce the fungus to produce the mycotoxin as a kind of defense mechanism. What substrate? Usually they need something fairly proteinaceous to grow on, something high in protein, carbon dioxide levels, how long something's sitting around, if there's any mechanical damage to the plant or insect damage. And something that's very important to remember is that organic crops can be particularly vulnerable because they're not always sprayed with that fungicide. So that's the prevention aspect, but also there's a number of places in the food chain, in the food production chain, where we could detect these mycotoxins and redirect product to avoid them ultimately reaching either these cute little kids or that cute little pup. You know, we don't want a contaminated product to reach the consumer, whatever they may be. So we talked about what mycotoxins are, why they're bad, why are they bad in terms of financial implications. What happens if something has gone through that whole production chain from growth, storage, you know, harvest, storage, processing, all of that, and it's shipped out for export and then it fails testing at the port? Well, you know, regulations are only as good as their enforcement, and one of the groups of nations that are particularly good at enforcement and at regulations is the European Union. The European Commission has some of the strictest regulations in the world, and you want to ensure that you don't fall victim to that. They have this rapid alert system for food and feed, which means that if a shipment is received at a border port and it goes through the sampling and testing process and they flag something that's high aflatoxin, whatever it may be, they notify all the other nations within the European Commission, and in fact a couple of others that participate in this same system. So you can't just turn that ship around and go off to the next country and say, oh, I'm just going to access this lucrative market here instead. It doesn't work like that. They really make sure that it can't. And these are just some examples from a couple of years ago. Germany is one of the major ports, Hamburg, one of the biggest ports in the European Union. And the top 5 hazards that they put notifications into the system for in 2018 included both aflatoxins and ochratoxin A. So that's two of the most common mycotoxins in those top 5 hazards. And of those, the top 5 product categories that get hit the most often with one of these notifications include things such as nuts and nut products, seeds, fruits, feed materials. So these are all things that you may be considering exporting yourselves. And this is, like I said, really a global problem. There's nowhere that is exempt from this. The European Union has the strictest regulations. Japan also is very, very tight. And there are multiple markets that can be affected by mycotoxins. I've listed quite a few of them here. They include the ones that we all know about, like tree nuts and peanuts and grain, but something that's growing and is a growing problem, cannabis products and botanicals, spices. We don't even think about that much. We use such a tiny amount, but if we're using that tiny amount over and over again, you have to think about that bioaccumulation. And we've got to think about our pets as well. I'm currently in my guest room and my cat is lying next to me, and he looks very peaceful, and I would be devastated if something happened to him. So I want to make sure his food is as clean as my kids' food. Animal feed, not just pet food, animal feed as well. So those are some of the markets that we are talking about, and things that you wouldn't even think are a food, like cottonseed, often go into animal feed, and they can also be impacted by aflatoxin in particular, which is a key factor for the dairy industry. If cows consume aflatoxin contaminated feed, it gets converted to another form of aflatoxin, which is excreted in their milk, gets into the dairy chain. Dairy is used in so many food products. It really is something that can have such a huge impact. And because it's such a global situation, there's all these different regulations from different countries, and it can be utterly overwhelming trying to figure out which test you need to be doing, which regulation you're trying to meet. So we actually have this really helpful website called the commodityregs.com. If you go on there, you can select the country that you're considering, the commodity that you're looking at, and it will tell you what regulations you need to meet for which mycotoxins. And I think from there, you can figure out what tests you might need to do. And just to think about what these regulations are, for something like aflatoxin M1, the European Commission sets a maximum limit for that of 50 parts per trillion. For aflatoxin B1, it's around 5 parts per billion, ppb, for many products. And to just conceptualize what that means, looking for 1 part per billion is like looking for a single pinch of salt in 10 tons of potato chips. So, I mean, I love potato chips, but not that much, So you're going to need something that's a very rigorous test that can reach those limits. It's not easy to find. What it comes down to is if you put rubbish in, you're going to get rubbish out. If you use a rubbish material, you get a rubbish product at the end. If you use a rubbish test, you get a rubbish result, and that can have an impact on your product. So how do we find that needle in the proverbial haystack? How do we find that pinch of salt in the 10 tons of potato chips? How can you protect your product? So the first step is sampling your consignment. This is the biggest challenge in mycotoxin analysis, is the biggest source of error in mycotoxin analysis is the sampling step right at the very beginning. Mycotoxins are known to be often unevenly distributed throughout a consignment. And you want to be able to not only detect isolated hotspots of contamination, but also to consider the whole lot. Just because there's one bad nut, you don't want to write off the entire consignment. So if you have a good sampling procedure, you're at the best place to start your testing. So what you really want to do is take incremental samples throughout the lot, throughout the consignment, and combine those. And actually, the European Commission, again, lays out a really good protocol for this. They've done a lot of work on it. And it's published online. You can look that up and follow it. It really makes sense whether you're at a port or whether you're back in your lab at your peanut processing plant. Wherever you are, you need to think about how you're sampling your consignment, because it really is impossible to assess what further action you need to take if you find contamination based on the analysis of an unrepresentative sample. It's meaningless. And this is such a big challenge that people have written entire books about it. I don't recommend reading them, really boring. But that European Commission website is very helpful, and it is formal guidelines. And just as an example of how this can have an impact and is having an impact, this is the Japanese fig conundrum. Japan's aflatoxin sampling and testing protocol is different from that used by the USDA. They have a more stringent action limit, so it's, it seems better. At 10 parts per billion, a lot of dried figs or nuts would fail. And that's a lot lower than the United States, which I believe is 20 parts per billion. But the difference is that they don't allow averaging of test results. They don't allow you to rework something if your results look odd or fail. They don't allow for retesting. And their method of sampling really increases the likelihood of hitting one of those hotspots, but not considering the rest. Instead of taking many incremental samples, they consider much smaller amounts. If we talk about this a little bit more, I think on the next slide, they start with a 10 kilogram sample, which seems fairly substantial. 10 kilograms of dried figs is about 580 to 600 pieces of fruit. And if you compare that to 10 kilograms of something like almonds, which can also be problematic, that's 8,000 nuts. So if you've got one bad fig in 600, that's a lot different than one bad almond in 8,000. And that shouldn't really be a problem if you then process that sample properly. So with figs, they're big and they're sticky, and it's very hard to homogenize it, especially if you just use the whole batch of figs. If you've ever tried chopping them up, they stick to the knife. You can't just blend them into a paste very easily. You have to blend it, push it through a sieve, blend it some more. But really, what a lot of people use is a slurry, where you add water while you're blending before you start your extraction process. And that really helps to homogenize that sample. But in Japan, the Ministry for Health and, I can't remember what the acronym is, the MHLW method and guidelines, not only have that odd sampling method, but they also simply state crushed and homogenized sample. They don't specify how you should crush and homogenize these figs. There is no slurry made. And what that ends up doing is that you may get these strange results that are a little high. You might pick up on those hotspots. You're going to get a lot more variability, and it's going to be a lot less reproducible. And so people are running into this problem that they're sending out consignments of goods all the way over to Japan, and they're failing upon entry. The best way to defend against that is to make sure you're testing well before that export point. And so I think now we're going to talk about what some of those testing options might be and how you might figure out what you need, so you don't run into that problem where at the point of entry to your market, your product is failing. So there are a full spectrum of testing options out there. They run from the most simple dipstick test, very cheap, very quick and dirty, all the way up to the highest end instrument, the most fancy thing you've ever seen on TV, in the background of some science show, and then everything in between. So we're going to talk a little bit about how you choose which one. Where do you fall on that spectrum of complexity? So I think one good place to start is to consider the golden triangle. You've probably all seen this before. It applies to many situations. The idea is that the three sides of the triangle, you can have something that is very fast, that works rapidly. It's going to get you the result you need in the shortest time possible. You can have something that gives you really good results, really reliable, really reproducible, really sensitive. And you can have something that is cost effective or cheap. You're watching your bottom line. But you can't have all three of those things at once. You can only choose two at a time. But luckily, when you consider this technology pyramid in the center of the slide, there really is something that covers every side of those triangles. So the majority of testing is taking place at receiving points, processors early on in the food chain, maybe some commercial labs or transit points. I'm thinking of peanut buying points, or a grain elevator where the trucks are waiting in line. So you need something that's very quick, very rapid. That's where something like a lateral flow strip test or an ELISA or a fluorometer test can often work. But then you start to move up in complexity, and you consider official government labs or commercial labs that are serving people that, or companies that are looking to export or sell to a large market, and they're considering something that's a more analytical test that is maybe compliant with regulations. So that's when you're thinking about something like liquid chromatography, be that HPLC or UPLC, and then all the way up to something like LCMS, which is the most fancy and most expensive. So just to go into a bit more detail as to what those options really are, lateral flow tests are very simple. They can be as simple as a dipstick, a bit like a pregnancy test. It's the same technology. Are you pregnant or are you not? Do you need to panic buy diapers or not? Is my product contaminated with deoxynivalenol or not? Does it exceed 20 parts per billion of aflatoxin or not? But maybe you want a bit more detail, and lateral flow is improving all the time. Right now it can be combined with a fairly simple and inexpensive reader that can quantify. So it reads those lines and compares it to a calibration, and it can give you a quantitative value of exactly how much aflatoxin is in the extract that you added to the strip. And you can make your decision there based on this great screening tool. So they're usually in a competitive immunoassay format. Immunoassay is just another fancy name for lateral flow or ELISAs. They can give you quantitative, very sensitive results. For example, I think I mentioned earlier that the European Commission maximum limit for aflatoxin M1 in milk is 50 parts per trillion. So we actually have a lateral flow test that goes down to 25, a limited detection of 25 parts per trillion, which is a very abstract number, right? But if you think about that pinch of salt in 10 tons of potato chips, it's 40 times less than that pinch of salt. Like, I don't know, very, very small. And they're very quick. You're going to get your result in less than 10 minutes generally, which is great if you've got those trucks waiting in a long line. They're very, very easy to use. The guys that are driving the trucks can probably run the test. That's the way that they're generally designed. So they're excellent for screening at the point of collection. And as I said, they're improving all the time, and very often they can meet worldwide regulatory limits, so you know what you're sending out there. But unfortunately, or fortunately, depending on your perspective, those regulations often dictate what type of testing, and something like lateral flow may not be sufficient. It may be considered only semi-quantitative. So you need to step it up a little bit, which we'll talk about in a moment, just to give you some real results of how lateral flow can work. This was a multi-toxin, couple of multi-toxin naturally contaminated corn samples that came into our lab. They were tested, a single extract was made. It was particularly nice. It's non-hazardous, no solvents used. So single extract, it was tested for the 5 different mycotoxins on that same extract. And the results from the lateral flow test very closely correlate and match the HPLC results for those same corn samples. And it's also pretty reproducible between the 3 test strips. So it's just a little bit of snippet of data just to give you an idea of how reliable these can be. But like I said, if you're trying to meet some regulation, you might need to step it up a notch, or you might want to look for further detail on your results. And if you're going up to a fluorometer, a liquid chromatography, or a mass spectrometry option, you may need to consider some sample preparation or cleanup. And the 2 most common methods used for cleaning up an extraction before injection into one of these instruments is either SPE, a solid phase extraction, or an immunoaffinity column, IAC column extraction. And what those do essentially is to wash away all the extra gunk and stuff that's in your extract, all those plant proteins that make things delicious but really mess up an instrument. So you can see in these illustrations, there's a number of different shapes going into the 2 different types of columns. And in SPE, they're just kind of sorted out, and you rinse away what you don't want, and then you elute what you do. With an immunoaffinity column, you have something called an antibody in there on some kind of matrix, and that works kind of like a lock and key or a hand in glove situation. Those antibodies will only grab your analyte of interest, and they hold on to it while you wash away everything you don't want, and they hold on to it until you elute them. So what you end up with something that's very clean, and in the case of an immunoaffinity column, also can be concentrated, which is very useful when you're looking for something at a very low level, or you're expecting that you won't find very much, but you really want to know what's there. So you have this elution, this eluate. What do you do with it? How do you choose where to go next? Are you going to go to fluorometer? Do you need something really high level and go to mass spec, or can you settle for something in between? What do you have available to you? Well, you know, they're all viable options. If you use it with a fluorometer, it's again a fairly inexpensive, easy to use instrument. It can be used in a field environment or in the lab, and it's been established for many years. It's widely accepted as a method by the AOAC and USDA. And again, it's quantitative. You'll get a number, generally with a printout. And it's very good for sorting products. And many of these columns don't need to be refrigerated, so you know, it helps you with storage issues, and they're pretty rapid still. You can still get results within 7 to 20 minutes. And something that's nice, that's slightly different to a lateral flow test, is that you'll get a much wider assay range, up to 100 parts per billion. And just to give you an idea, again, of a little snapshot of some real results, this was a collaboration we did last year with a customer, Golden Peanut and Tree Nuts. They were interested in streamlining some of their testing that they had going on at their buying points. So we developed a method that would work for them, but they were concerned as to whether it would match up with their fluorometer testing and even their LC testing that they were doing at their central facilities to be compliant with export regulations and so on. And so what we did was they chose a number of samples from their regrade facilities, and the varying levels of contamination from very low to very high and a few in between. And they tested them down there by fluorometer, and to establish whether they were contaminated or not, they sent them up to us. We tested them by fluorometer and UPLC, and also our AFLA-V lateral flow test. And what you can see is that the fluorometer very, very closely corroborates not only the lateral flow test results, but especially important, the UPLC results. This was with a well homogenized sample, something, again, that sampling is very important, that sample preparation is very important. And what happens is that immunoaffinity column cleanup really gets rid of anything that can have, well, a lot of things that can have an impact on your results. And so the fluorometer and UPLC of that R squared value is, you don't really see much better than that. So again, you know, maybe you want something more than the fluorometer. The fluorometer can't, for example, break apart the various forms of aflatoxin. There are 4 common forms, and they could all be present in different levels. And in some markets, specific forms are regulated. So for example, off the top of my head, Canada, in cannabis products, they specify a specific amount of aflatoxin B1 that can't be exceeded. So you couldn't identify that by fluorometer or lateral flow. So you're going to need to step up your level of instrumentation and your level of training. So you could use immunoaffinity column cleanup with UPLC. This is an official method of the AOAC and CEN. It's an accepted method for exports. It's considered a confirmatory method. And there are many methods available out there for many different types of samples. It's been around for many years. And as I said, you can quantitate individual toxins, whether that's aflatoxin B1, B2, G1, G2, or the fumonisins also have 3 different forms. And depending on your level of instrumentation, you could perform a single toxin analysis. You could do something like a Pourcy toxin analysis, what we're talking about here with just a few analytes. Or even if you have a particularly fancy instrument, you can go all the way up to a multi-toxin analysis, which we'll see in a little bit. And the nice thing about this is that these instruments are very expensive, but you can use them for other forms of analysis too. You can reach lower limits of detection, and they integrate nicely into your laboratory environment. So here's some real results. This was afla-ochre testing with an immunoaffinity cleanup. I believe this was spiked flour. This was during a method development that they were doing at New Mexico State University. And this data has been corrected for recovery, but you can see that the recoveries are really looking great. This is what he was looking for. Cannabis and cannabis products are particularly difficult to test because they're very complex. They have a lot of different oils and other pigments in there. So that was that one. And you can see this is a much simplified view of the same information. You can see really clean, distinct, and easily identifiable peaks for the different toxins compared to an unspiked sample. That's after cleanup. You wouldn't see that without cleanup. And this is an example now of something with a mass spectrometer, so much more complex, much more advanced. You're going to need highly trained scientists to do this, but again, using immunoaffinity column cleanup, you can get results for, I think in this example, it's 12 mycotoxins all at once with great recoveries. This work was done by a collaborator from the Chinese FDA who wanted to develop a quick, relatively quick analysis for multiple mycotoxins in traditional Chinese medicines. And the example that she used for her model system is illustrated in the picture at the bottom of the slide. This is something called astragalus root, or huangqi. I'm probably saying that wrong, but I think it's huangqi. And it's one of the most widely used traditional Chinese medicines in the world. This is a really growing industry. And it's something that China's taking very seriously. So they wanted a robust method. And you can see, I can't even fit all the analytes on one slide. But again, very clean, easily identifiable peaks. But all of that can be overwhelming, right? So if you're not a scientist and you're not a lab rat like us, how do you know what you're doing? If you don't know, find someone that does. Talk to someone. There are plenty of programs out there that verify labs know what they're doing and are using methods that are trusted. For example, the USDA has a program where they inspect and approve laboratories that they deem good at what they say they do. And this was initially set up for pre-export testing at the time, I think for peanuts, although that's since expired, but now for almonds and pistachios, as well as a number of other things. And so they go through this annual inspection every year and make sure that they can get the real results that they say they can get. And they receive this approval. And the list of labs that have those approvals is available online, very easy to get hold of. This one I pulled up the other day, you might recognize these guys, familiar name. So yeah, the Safe Food Alliance is on there. It says which test methods they're comfortable using, what they're approved of, the people that are signed off to do that. So they're usually very happy to talk. We love to talk too. At Waters, we also take part in proficiency testing. The example badge there was for, I think it was cannabis, the Emerald test, testing for mycotoxins. So with that, I'll just go over our conclusions from today. I think we can all agree that mycotoxins pose a major risk to human and animal health. Many commodities and markets are affected, even those that you wouldn't expect. And it really is a global problem. And it can be very hard to pick your way through that minefield of regulations that can be overwhelming, but it's not impossible. And there are people out there to help. Prevention is always better than cure. You can probably tell from my accent that I'm European, right? And over there, we firmly believe in the precautionary principle. So detecting early, preventing it, all of that is better than trying to fix things afterwards. That can protect your business and your product. And so if you're testing frequently with the appropriate test, that really provides safety for your profit margins. And no matter what you're doing, there's an option out there that will meet your mycotoxin testing needs, whether it's something, you need low cost, quick, simple, on-site, you just want whoever your tech is to be able to run the test. Lateral flow is a great option. Fluorometer analysis with sample cleanup is accepted as a regulatory compliant solution, and it's a very cost conscious option for smaller testing facilities. Porcine mycotoxin testing, so perhaps testing for aflatoxin and ochratoxin, as we saw in that example of afla-ochra, that can be best met with immunoaffinity column or SPE cleanup and an LC system that can reach low limits of detection with excellent sensitivity. Maybe you're in an environment where you're looking at research, you're trying to meet very low levels. I'm not sure what your situation might be, like the scientists at the Chinese FDA that we worked with. You might be interested in multi-mycotoxin analysis, and that's best met with some cleanup followed by LC-MS. So that cleanup not only protects your instrument from all the gunk that can go in there, but it can also improve your results and help you concentrate those toxins down so you can reach some lower limits. So if you just have a think about what your testing needs might be or what your questions are, and talk to us. It says on this slide, visit us. Can't do that right now, but you can call us, and we're always happy to chat, and you can find us online. And just a reminder to pick your way through those regulations around the world. There is this tool available online. I highly recommend that website. And with that, Dr. Hall and I will take your questions. Thank you.
All right, great. Thank you, Elise. That's a very, very informative talk, really covering a lot of material on the world of mycotoxins. So, we've got a number of questions in. Just to kind of start off on some of the more general area questions. You talked about different, you had your list of different mycotoxins and different commodities and crops that might be affected. Are there areas, either in terms of the mycotoxin or in terms of the commodity, where you think that it's under tested, where someone in that industry might really want to get on it because there's maybe kind of a sleeping problem?
Right. Yeah, definitely. Like I said, I really believe in the precautionary principle, and I think if you can do some risk mitigation early on, you're going to be in good stead. So the European Union's been threatening for many years to regulate ochratoxin at a limit of 10 parts per billion in a number of products, I think including sunflower seeds, amongst others, hazelnuts maybe. And I think that's a really important one to watch. Ochratoxin, I think, is underappreciated for how harmful it can be. It's nephrotoxin, so it damages the kidneys. And it's in things that we consume a lot of, or a lot of us consume a lot of, like coffee. Like, that's our lifeblood at the moment. I'm in the middle of moving house, drinking a lot of coffee. And as I said, it can bioaccumulate. So I think testing for that needs to be taken more seriously than it currently is. I know the EU is also talking about regulation for ergot alkaloids that cause that ergotism, that syntagtanese fire that we saw in that strange picture at the beginning of the presentation. That's another one that's on people's radar.
Great, thank you. And again, your website, Commodity Regs, is a great source to kind of keep up on some of this information. Also USDA, FAS, and there's other agencies and commodity boards. You want to make sure you stay informed. I had a couple questions that came in about kind of where the contamination is. So this was someone who was concerned about spread of the mycotoxin contamination, either in storage or during processing. And I guess it's really going to change a bit from commodity to commodity. But can you talk about kind of where on a given piece of fruit or grain or something where we find the mycotoxin?
Sure. Yeah. So as you said, it varies by commodity. I think one thing that's a common misconception that is important to understanding this is that the mycotoxin itself is not alive. The mycotoxin can't grow. It comes from the mold, and the mold doesn't produce the mycotoxin in all situations. But once it does, it's there and it doesn't go away. It's almost impossible to eliminate a mycotoxin once it's present. So you have to redivert that. Now, the question as to whether it can contaminate adjacent product within the same lot, yes, it can. Sometimes it's found on the surface, the mold grows on the surface. It can also be inside in the endosperm of the grain or nut or whatever it might be, or fruit. If you think about some of the early splits that can happen in nuts or insect damage, the mold can wheedle its way in and the mycotoxin can be in, or maybe it's just on the shell on the outside, but you can't assume that it's only in one place, which is why sampling and sample preparation is so important. Something like a soft fruit, like the figs that we talked about, often grown in warm, humid climates, that mold easily spreads. So if something is not stored at the proper temperature or the proper humidity level, it's not dried out quickly enough after harvest, the mold can spread and consequently the mycotoxin can spread. And again, it can be on the inside as much as the outside. Nobody can really separate that out. There are, considerations that if the majority of the contamination is on the shell of a nut, well, you can discard the shell, you don't eat that bit. But then you think about something like a pistachio, and we often put the whole thing in, right? And you suck the shell as well. So I think testing of the whole product is important. Proper mitigation is also really important, and that includes storing contaminated material away from your clean stuff.
I think the fig is a good example where, you have what looks like a perfectly healthy fig, but, you cut it open and, it's all mold inside, just from the way they grow. And that kind of leads into another question where someone was asking about, ways to remove the aflatoxin, or mycotoxin contamination. I know that you can go and, kind of maybe resort and hopefully get rid of those, moldy or, contaminated product. Or is there any, technology that you know of that can remove mycotoxin contamination?
There's many that try. Any that can, no. I think the thing to think about most mycotoxins is they're organic compounds, and they're quite stable. And a lot of the things that make food healthy, nutritious, and tasty are also organic compounds. So anything that's going to destroy a toxin is potentially going to destroy the food product, and make it completely inedible and useless. So anybody that can come up with some kind of mycotoxin mitigation thing that somehow eliminates it afterwards is going to be a very rich person, but many have tried and many have failed.
We were talking about it before the webinar, but we had a recent project where they, someone had a technology that was great at destroying aflatoxin, but our lab manager was getting upset with me at the quality of the nuts was just destroyed, and she was pretty mad at these horrible nuts I was making her eat for organoleptic testing.
Yeah, that doesn't sound fun. I wouldn't want her job.
So, no. So to get into testing, we have a question from someone who's concerned about the best way to remove color from their extracts. This is specifically black tea and composite flours that contain sorghum and millet, and they're using an ELISA method. So do you find that a heavily colored extract will interfere with being able to read the results well?
Yeah, often, especially with something like an ELISA, it's essentially a colorimetric assay. If you can't wash that away and eliminate it, it's a problem with lateral flow tests where whether you're doing a quantitative test or a qualitative test, those little lines that you're looking for, they're usually red, which is a gold conjugate. And they can be very hard to see if there's a lot of pigment. Dried distiller's grains is something else that tends to have a lot of pigment and can interfere with the results. So with something like that, you're going to want to use a good cleanup beforehand. I would recommend switching to something with immunoaffinity column cleanup and fluorometer or LC, just to get away from those compounds and pigments that are interfering with your result, because it's very hard to do that without a proper extraction and cleanup step.
And of course, the benefit to going to that immunoaffinity column is you'll have the concentration of any contamination that's there, so you'll improve your detection limits as well.
Definitely, far more so than ELISA. Yeah.
We had another question just come in that kind of goes to the last one we were talking about. So you had alcoholic beverages listed as industries that might be concerned, and you mentioned how you can still have the aflatoxin M in dairy products, if the cow eats something that was contaminated. Do you find do you know if there's any reduction in mycotoxins from fermentation or other types of food processing?
So generally, no. In fact, wine it can be at risk from ochratoxin contamination from the Aspergillus species grow quite commonly on grapes used in wine production. And what we found in a lot of our work with the cannabis industry is that some of the extraction techniques that they use, distillation, can actually concentrate the mycotoxins in the same way that it concentrates the compounds that they are interested in. The beer industry is very conscious of contamination with DON, that vomit toxin that I mentioned. And so there's multiple testing steps throughout their process to try and eliminate that. Contaminated barley or wheat or hops can introduce mycotoxins into that process too. And as I mentioned, dried distiller's grains, which are a byproduct of the ethanol industry that are actually used in, I think, pig feed as well as other animal feeds, can be very, very highly contaminated with aflatoxin.
These are really kind of almost famously stable compounds, and a lot of the things that we might think of as that would destroy or remove contamination,
right.
Washing, drying. Maybe if we're talking about dried fruit, you're only going to increase the levels that you get.
Right. Yeah, it really does. And dairy, when they talk about powdered milk, it's used in the production of many different things, from bread to different cheeses and yogurts it's added to. Again, that concentrates down any toxin that's there. It's really, really hard to get away from. And we talked about how it can be on the outside. And okay, something like aflatoxin is not so stable under UV light. But what if you're not just on the outside, you're on the inside too? Then there's no way to know, and there's no way that light's going to get there. So preventing the production of them in the first place is the first step, and rigorous testing is really the only way to protect yourself. So
if I'm running, say I have, say a mill or a packing or processing facility, and I want to, like you say, kind of get proactive with things and do some testing on my own. For the various test methods that you mentioned, what type of training or background does the worker or whoever is doing the analysis need?
So that depends on the type of testing that you're doing. As I said, many buying points, grain silos, places like that are using very simple rapid tests, either lateral flow or maybe the fluorometer test. Lateral flow, those tests are very, very simple to use, and a lot of the people running them may not have a high school diploma. they're designed so that they can be run in a tin shed in a field in Georgia, or in a fancy high-end lab in a big city somewhere. They're very straightforward and easy to use and affordable as an option. Something like immunoaffinity column cleanup with the fluorometer is slightly more complicated, but there are people out there who assist with training. I know we send out teams all over the place to help people get that going in their facilities, and it's often a great way to get regulatory compliant testing, confirmatory testing that's accepted going in a facility that perhaps doesn't have vast amounts of resources, or only has one dedicated scientist who perhaps they want to put on their fancy high-end instrument because they need to focus on their pesticide residue testing or whatever it might be. And you can have a technician with less training running the fluorometer mycotoxin testing, no problem.
So I see we're just about at the end of our time. So maybe to kind of wrap things up, I'll ask you to break out your crystal ball and what do you see as the kind of the future for the regulatory environment and the testing that goes along with it with mycotoxin?
I think we're going to see more regulation. I think I mentioned ochratoxin regulation is coming. Ergot alkaloids are coming. I think as we see more climate change, the environment changing, different mycotoxins are going to pop up as a problem in areas they weren't before, because those fungi are so susceptible to their environment, be that the weather or the storage conditions afterwards. I think we're going to see changes in regulation in developing nations as their food production systems mature, and yeah, I think there's going to be more, and I think we've all got to be proactive and try and mitigate those risks before it's too late, before those things hit and we have no choice.
As a brief bonus question, with the UK and Brexit, have the regulations changed compared to when you were shipping to Britain when it was part of the EU?
Who knows? That is a very good question. It is such a mess
yeah.
over there. So I talked about the rapid alert system for food and feed that the European Union has, and the UK used to participate in that. I believe as of now they still are, because nobody can figure out what they're going to do instead. They did build the framework for very similar rules and regulations and system for importing food. But again, I don't know how it's going to work with Ireland and the border there. It's a big mess. And I just hope that for the sake of my friends and family that are back there, that everyone that's exporting to the UK is patient and still continues to test as if they were part of the EU, because I want them to be safe.
All right, great. Well, I know that you're moving right now, and I don't want to keep you from the joys of packing and carrying things out any longer than we have to.
Are you sure?
I really want to thank you.
I quite like to hide in here.
I do really want to. Oh yeah, but I really want to thank you for taking the time to kind of talk our audience through some of these issues. And with the add in that if you had a question that we didn't get to, please send it to us by email, and we'll either try to answer it ourselves or maybe send it to Elise, and we'll try to get an answer back to you.
Absolutely.
But I want to thank everyone for joining us today, and everyone have a good day.
Thank you. Bye-bye.